That looks good. That looks good. Welcome, everybody, to Plato, Frontiers in Life Sciences. This is Dr. Grace Wilkinson, who agreed to do this about 2.5 weeks ago when a news release came out. I hope you saw the story in the State Journal yesterday. So one of the great things about science is having to go with the data, in my opinion, especially when it isn't what you expect it to be. So I hope it's a pretty cool story. We're now going to ask you some questions about yourself. Where were you born?
Yeah, I was born in St. Paul, Minnesota.
Oh, up north.
Yeah.
Jeez. Just east of that, what's that other place? Mini no place. And then where'd you go to high school?
I went to high school in White Bear Lake, Minnesota. Go Bears. Yeah. So up in the northern Twin Cities. Absolutely.
And then where'd you go for your undergrad? What'd you study?
Yeah, so my undergraduate was at St. Olaf College, which is in Northfield, Minnesota. It's an undergraduate only institution. And then I got my PhD at the University of Virginia, but I did most of my research during that time in northern Wisconsin.
Wow. What was your PhD research?
Yeah, so I'll actually talk a little bit about some of that work today, but my PhD research was on how much carbon from the land comes into the lake and supports the food web in the lake. So essentially our fish made of trees.
And now you're on the faculty here at UW-Madison.
Correct.
And you're in the Center for Limnology. And what is your home department?
Yeah, so the Center for Limnology, and then I'm also part of integrative biology.
Great. Well, thank you. And go ahead and take it away whenever you like.
Wonderful. Thank you so much. And I appreciate the opportunity to come and speak with you all today, both here in person and online, about some of the current research that's happening in my lab right now. And if at any point I'm not being loud enough or whatnot, feel free to just raise your hand and let me know. Yes.
So right now I'm seeing your next slide. Like that.
Oh, that's no fun. Thank you for letting me know.
Let's see what we can do to adjust that.
So it's not next, that's just the next slide.
Not next to us, not next to you, not next to you. Just me.
How about now?
Yeah, I'm still seeing that.
Right, it's because it's not sharing the correct screen. So one moment, we will fix that.
Oh, so you're far more confident. I'd be saying, what the?
All right,
Buttons randomly.
that should hopefully solve our problem.
That looks great.
All right, wonderful. We'll get the wonderful green border. Lets us know we're on Zoom. Wonderful. Well, I'm excited to share some of the current research that's occurring in my lab right now that's really sort of on the topic of algal blooms. And you know, if we open up the local newspaper, turn on the TV, or even on social media, it seems like more and more we're hearing about harmful algal blooms. They're likely in water bodies that we love to recreate on, perhaps lakes like Mendota. And as a person who loves lakes, like I said, I grew up in Minnesota and around a lot of lakes. I love lakes both as a person, as a scientist. When I see these stories, I tend to ask questions like, what's causing that algal bloom? Is it getting worse? Is there something we could do to be better stewards of that freshwater resource to try to prevent this from happening in the future? And these are some of the questions that not only I have as a person, but underpin the science that we do in my lab. And so today I was hoping to tackle these 3 questions. First, we're just going to talk about what are algal blooms and what causes them. Are algal blooms getting worse in lakes? And then finally, if a bloom is going to happen, can we predict it before it's going to happen in an effort to perhaps mitigate the consequences of that bloom? So to start, let's just all make sure we're on the same page. When I say algal bloom, what do I mean? What is a bloom? So blooms are a rapid proliferation of phytoplankton, so that microscopic green algae, like you're seeing in this picture here, that grows to a nuisance and sometimes even a harmful level. And there's many reasons why we care about algal blooms and think about their consequences and why they might be harmful. But one that's most important, perhaps to us as humans, is the fact that cyanobacteria often dominate in these blooms. And some genera of cyanobacteria produce toxins. And these are toxins that act upon the liver, as well as neurotoxins. And if we're exposed to them long-term, there can be chronic effects, things like an increase in liver disease. However, there's also acute effects, right? So if you take a big swallow of water, out water skiing and fall down, like, I don't know, maybe you're Tommy Thompson, right? And you might get a big gulp of water, and that can cause things like gastrointestinal distress, dermatitis, and other issues as well. These toxins are usually not in high enough concentrations to kill immediately, although for smaller animals they can. We see a lot about dog deaths for dogs that are splashing around in these waters. But they certainly can have very negative health consequences. And so algal blooms are not a thing we want in our freshwater ecosystems, right? They're something we want to prevent. So in addition to those potential health consequences, algal blooms also negatively affect ecosystem services. So these are the benefits that we get from lake ecosystems that we are not necessarily paying for, but they provide us with those benefits. So for example, algal blooms can interrupt if that lake or reservoir is a drinking water supply because of those toxins that I talked about. And blooms just smell, right? And so when you have really stinky water, water managers don't want to use that water for the drinking water source. It can interrupt food supplies through fish kills and also those toxins. When those toxins are at really high concentrations, or really the lake just looks green and scummy like you see here, it impedes our ability to recreate on that water body, causing things like beach closures, and it also just decreases the perceived value of that ecosystem as a place to go and spend time. It also decreases biodiversity, again, through things like food kills. It can cause a collapse of the food web or the organisms that live in that lake. Properties that are around lakes that experience frequent harmful algal blooms actually have a lower property value than they would otherwise. And overall, algal blooms result in nationally billions of dollars of economic loss every year. This is both considering blooms in lakes, so freshwater, as well as in the coast, right? So billions of dollars in economic loss. So what causes these economic catastrophes in our lakes and reservoirs? What causes blooms? Well, one of the ingredients you need for an algal bloom is a lot of excess nutrients, in particular nitrogen and phosphorus. And we refer to any nutrients that come from outside the lake in the watershed as being external. So it's external loading of those nutrients. And I also want to note here, it's not just what's running over land and coming in streams. There's also nutrients in our groundwater. We think a lot about sometimes high nitrate concentrations in our groundwater. That's a nutrient, right? And so nutrients are also transported there. But there's also already nutrients stored in the lake itself. And one example of that is the massive amount of phosphorus that's stored in the sediments of lakes and reservoirs. So in a lake like Mendota here, there's so much phosphorus down in the sediments that is from outside in the watershed. It could have come into the lake 20, 30 years ago, but it's just waiting to be released like a ticking time bomb of nutrients. And if we were to deal with all of the nutrient inputs from the watershed now, that source of phosphorus in the sediments is still going to cause problems for decades to come in terms of algal blooms. So it creates this lag. And also, it matters when these nutrients come into the system, when they arrive. If they're coming in the late fall, like now, there's not a lot of light, it's cold, not a lot of algae is going to grow. If it's the middle of the summer, more algae is going to grow, right? So seasonality, how much, and in what form. All of these things matter. How long those nutrients stick around or their residence time matters, as well as how much light they're getting, how warm the water is. Algae like it warm, just like us, right? And how many organisms are there to eat the algae or graze. And one of those organisms that I'm showing there is a zooplankton. It's a Daphnia, and Daphnia you can think of as kind of being like cows in the lake. They're really good at grazing on that algae. And then finally, how close that ecosystem is to what we refer to as critical thresholds or a regime shift. And I'll visit that a little bit later in the talk and what I mean in terms of regime shift, but how much resilience is in the system also matters.
do we ever deal with the food web control and organisms that are eating algae? So we can enhance them, or isn't that a good idea?
Yeah, that's a great question. There has been a lot of work to do just that, to enhance the food web control on algal growth. And it certainly works, but it works best when it's done paired with things like stopping nutrients coming in from the watershed. And that's work that's actually happened on Lake Wingra and Lake Mendota as well. But as you can see from this picture here, there are complex interacting drivers. They're both within the lake and coming from outside. And human activities are actually causing really large scale changes to these drivers. So as an example, we're seeing globally widespread eutrophication, so an increase in nutrients in our inland waters. This is just an example in the United States of total phosphorus inputs into the watersheds or the terrestrial environment. As you can see, the agricultural Midwest, there's a lot of phosphorus that we're putting onto the land. And this is coming from things not only like livestock waste and fertilizer, but also things like human waste. And there's actually so much extra phosphorus that this map is showing its likelihood or what has the potential for running off into lakes and reservoirs in our surface waters. As you can see in some areas, particularly here in the upper Midwest, I was most recently working in Iowa. This is a huge problem. We have a lot of phosphorus coming off the land and into lakes. And it's not just, that's measuring the phosphorus on land. Is it actually getting into the lake and doing something? We have pretty good evidence from surveys that the Environmental Protection Agency has done over the years that we're starting to see more and more streams and lakes that have higher and higher phosphorus concentrations. And what this figure is showing here is the percentage of streams or lakes in the United States that have a phosphorus concentration where we might start getting a bit concerned about algal growth. And as you can tell in the streams that they measured from 2004 to 2014, almost 100% of those streams have enough phosphorus in them. And our lakes are certainly getting there as well. So widespread eutrophication. Yes.
You know if there's what changes have happened since 2014?
Yeah, so this is a survey that happens every 5 years. So it happened again in 2019, and they're about to release the data. It happens on thousands of lakes and streams across the United States. So it takes a couple of years to get all of those samples analyzed. So I look forward to seeing what's going to happen next. Yeah, it's a great question. We're also seeing globally a rapid warming of our inland waters. So what this map is showing is everywhere where a lake has been measured over time and what its trend in warming is. And if it's red, that means the water is getting warmer. And in particular, you can tell we have good representation in the Great Lakes region up here, and our lakes are generally getting warmer. Algae love it when it's warm. What's more, precipitation is also changing. And in our area, precipitation is intensifying. In other words, those big storms, we're having more of them, right? So more events like that giant rainstorm in 2018 that occurred here, we're seeing storms similar to that, not of that magnitude, that was a big one, right? But similar to that more often. So precipitation is intensifying during the summer. And you can see that as a trend across the United States, but it's not the same. There's also some places like in the desert Southwest where it's getting much drier.
So we're not only having heavier storms, but we're having more precipitation?
Actually, that's a good question. Let's look at it for Wisconsin. Thank you. That was a great segue. This is just annual precipitation in Wisconsin. And you can see, yes, it has also been increasing. So if it's green in the darker, the green color there is where it's increasing. The asterisk indicates that it's been a significant trend over these from 1950 to 2018. So yes, not only are we seeing more intense storms, we're also seeing more precipitation. There's also been a change in our temperatures here in Wisconsin, right? So we've been thinking about globally and in the United States, but thinking about just in Wisconsin, what I'm showing here are the minimum temperatures. So we're not getting quite as cold during the day on average, right? That's a long-term significant trend. So things are definitely rapidly changing, and they seem to be changing everywhere, right? So given the fact that things like external loading of nutrients, because we have more nutrients on land, Precipitation is bringing more of it in. It's getting warmer, and we're also having more nutrients coming in, which is increasing that internal nutrient release. It would be reasonable to think, right, that blooms are getting worse everywhere. That would make a lot of sense, right? And in fact, scientists, limnologists have been assuming this for a very long time, and that this is what is occurring because we see these drivers changing. And what my colleagues and I wanted to do was say, well, we live in this beautiful time where we have so much data. People have been going out and taking measurements on lakes for decades. Let's actually go take a look at the data and see if this is true. In other words, are harmful algal blooms getting worse? And at the same time that we're seeing all of these drivers change, we're also seeing public awareness of algal blooms growing. What this map is showing here from the Environmental Working Group is that all of the newspaper reports of a harmful algal bloom in a local community that's happening. So this could be evidence that blooms are getting worse. We're seeing them more frequently and in more places. But what it's also probably evidence of is our awareness of blooms and the consequences and the potential health consequences are growing. And that's the thing we really wanted to tease apart. And so we simply asked, are algal blooms getting worse? And in order to actually answer that question, we first needed to define what do we mean by worse, right? So we came up with 3 metrics that I'm going to talk about today. The first is magnitude, and we define that as the average bloom in a year increasing or getting worse. In other words, how green the water is gets worse on average every year. Why do we care about that? Well, it's an increased likelihood that we're going to lose those ecosystem services that I talked about before. But it's not just sort of the mean or the average, right? We also wanted to look at severity. So is the peak of the bloom getting worse? In other words, instead of in the past going from what you see there on the left to blue to sort of greenish water during the summer, does it go from blue to the scummiest water, right? Is it getting worse? And when we have these more severe blooms, we have an increased likelihood of really severe consequences, things like summertime fish kills happening. And then finally, we looked at duration. How long is that bloom lasting each year? Is it lasting longer and longer? And there's a lot of evidence out there that the longer that a bloom lasts, or if that water stays green, that there's a perceived loss of value of that lake for going and recreating. And so if a bloom's lasting longer and longer, that increases the period where we could have summertime economic loss because of that bloom. And so these are the 3 metrics. These are the 3 quantitative things we wanted to look at to understand if blooms are getting worse. And before I dive into sort of what we found or how we went about doing that, you might be wondering, how do you actually quantify change in bloom, these bloom metrics? And there's a really great measurement that we use as limnologists. We look at the chlorophyll A concentration. So chlorophyll A is the pigment that's in algae. It's in leaves on trees and grasses, right? That's what makes it green. And we can extract that from the algal cells from a water sample and quantify it in the lab. And it's a pretty reproducible, quick, relatively cheap way to measure algal biomass. The other way you have to do it is look at all of those organisms under a microscope and count them and measure them and whatnot. It takes an expert to do it, right? So this is a really good way to do that. And that's why we use it so often when we monitor lakes. This is a tool measuring chlorophyll A that has been used on thousands of lakes across the globe for decades. So we can use this data set to answer this question. And what I'm showing here are some samples of extracted chlorophyll. You can see that the water there is somewhat green. That's because we've extracted the pigment out of it, made it dissolved. So now that we had our bloom metrics and we have a variable that we're going to use to measure this change potentially, we needed to make sure that the data were adequate to actually answer our question. And in particular, they needed to be frequent enough samples to capture that increase in magnitude, severity, and duration. Just as an example of that, this is some data that one of my students, David Ortiz, collected in Iowa. And David put a sensor out in the lake that measured chlorophyll every 30 minutes. So in other words, we can almost think about this as the true, right, measure of chlorophyll. At the same time, the Iowa Department of Natural Resources has a lake monitoring program where they visit all 130 lakes in Iowa, publicly significant lakes, a little bit different number than here in Wisconsin, right? But they visit all of those lakes 3 times during the summer and they measure chlorophyll, right? Because our resources are not infinite. You either have to measure very few times in tons of lakes or a lot of times in few lakes. So they choose to do many lakes a few times. And if we look at when the monitoring program went out and took those 3 samples versus David's daily data of chlorophyll, you can see that if we calculate these different metrics, they come out pretty different, right? The bloom doesn't look nearly as severe because we didn't capture that peak. The duration looks a lot longer, even though it was in fact shorter, and the magnitude in fact changes as well. So we needed to figure out where the sweet spot was between daily data, which pretty much no one has, and these really infrequent data. And long story short, we came up with every 14 days. There has to be a measurement made on the lake every 14 days in order for us to calculate these metrics. In addition to that, we didn't just want to look at the trends over a couple of years because we know there's a lot of variability, right? 2018 was this extremely wet year here in Madison. This summer it was pretty dry, at least at the beginning of the summer, right? So things are really different. We want to look at long-term trends, not these short-term little spurious waves. And so in order to do that, we said that we had to have at least 10 years of data where measurements had been made every 14 days during the summer. And just to quickly illustrate why 10 years of data, these are data from the North Temperate Lakes Long-Term Ecological Research Program, which is housed at the Center for Limnology. And measurements of chlorophyll have been happening on lakes up in northern Wisconsin and now down here on Mendota, Monona, and Wingra for 40 years. And if you look at, this is just showing the magnitude, so the average. If we just looked at a short window in here, we might think that there was a significant, very strong decline in bloom magnitude. We looked at another short window, we might think there's a pretty strong increase in bloom magnitude. But looking at the whole 40-year picture, we can see that there's not actually that much happening in terms of a trend, right? You need that long view of time. So what we were looking for were lakes that had at least a decade of measurements, at least every 14 days during the summer. And it turns out these were kind of unicorns. These weren't very easy data sets to find, but we did find 323 lakes that met these criteria. So we can ask for now over 300 lakes, are algal blooms getting worse? And if statistics is your jam, I'll just quickly mention that we were looking for significant trends, either increasing or decreasing. And these are the sort of nitty gritty of the methods that we use that I'd be happy to talk more about later. So to our question, are algal blooms getting worse? And before I move on, I just want to take a moment to acknowledge my 3 co-authors on this work, John Welter, Mike Pace, and Cal Bulow at the University of Virginia. As well as our funding from the National Science Foundation to complete this work. And that's the paper that just recently came out about this. So what did we find for these 323 lakes? Surprisingly, shockingly to us, we didn't find any evidence for widespread intensification of algal blooms. In other words, blooms weren't getting worse everywhere. What I'm showing here are the frequency histograms, so the 323 lakes and what their trends were, whether they were improving or deteriorating. But we really wanted to look at what those significant trends were over time, right? And only about 1 in 10 lakes that we looked at was getting significantly worse. So the hypothesis, remember, had been that this was widespread. This was 50, 60, 80% of lakes were seeing this, only 1 in 10. And very much to our surprise, in fact, 17%, so more lakes were getting better. Their bloom magnitude was decreasing. Their bloom severity was decreasing. The duration was decreasing. And so that really got us scratching our heads. First of all, this was not at all what we had hypothesized or expected, or what the narrative in our scientific community was. And so we wanted to understand a little bit more, think about why this could be the case. So why are so many lakes getting better? Well, it's not one thing that we went to is the literature to just try to understand, is this something else that other people have been seeing that we just haven't picked up on? And during this time in particular, Simon Topp and his colleagues used remote sensing, so satellites, to look at 14,000 US lakes. They couldn't measure chlorophyll, but they could look at water clarity. In other words, if there's a lot of algae in the water, that's not very clear water, right? And they also found that in 14,000 lakes, on average, water clarity is improving across the United States. So a completely different method, and that gives us a little bit more evidence or support for the fact that what we saw in our trends might actually be what's happening in the real world. And one thing that Simon Topp and his colleagues, and that we've also been investigating, is that perhaps what we're actually seeing is some benefits of the Clean Water Act finally coming to pass, right? So in other words, is the Clean Water Act and other water quality policy potentially at play here? And it turns out it's really difficult to find out what sort of funds from the Clean Water Act or local management, state management has been happening on a water body. Sadly, there's no, US-wide database where everybody inputs what they're doing in terms of management on these publicly significant water bodies. And instead, what we ended up doing was going to local newspapers, which when there's a big restoration that's happening on a lake or something that's planned, the Friends of Lake fill in the blank are donating money to do this. They run a story in the local paper. And we found that 65% of the lakes that we saw that were significantly improving had restoration efforts that were reported in the local news. So it was a really difficult, but I think kind of a fun way to find out whether or not there were restoration efforts happening on these water bodies across spanning 17 states and hundreds of lakes. Yes, you had a question.
Don't zebra mussels add to clarity?
Yeah, absolutely. Turns out nobody keeps an invasive species database either. And that is something that we are also looking at. And in fact, we're working on, this is our next phase of this project, is that exact sort of question. Can we pull together invasive species information, restoration information, a lot more about what's been happening in those lakes over time, so we can understand, I think about these as trajectories. The lake is on a bloom trajectory, and can we understand why this lake is on a given trajectory, the lake right next door seems to be on a completely different one. And things like invasive species might be a part of the answer. Absolutely. Yeah.
Some communities have lakes that are part of a watershed and have flooding problems.
Yeah.
So they've gone to the point of dredging and getting rid of sediment so that the watershed drains more efficiently.
Mm-hmm.
Would that improve or not?
Certainly that it could. And that is actually a technique that's also used in a lot of reservoirs, like here in the Midwest, to remove some of that phosphorus pool and to make it be deep enough where it's not going to be as accessible to the algae. And there's really mixed evidence about whether that actually helps, which is kind of part of that puzzle that we're trying to solve. Absolutely. So sometimes our best restoration efforts don't yield the best restoration results. So.
Well, I'm wondering whether dredging would actually reduce phosphorus.
Yeah, so I love these questions because this is exactly the things that I think about all the time. We do see some evidence that immediately following dredging, so for example, in some reservoirs in Iowa, that there's a very short-term spike in phosphorus, but it goes away pretty quickly. In other words, in a couple of years. But it is, and that's probably because it's changing the redox conditions. In other words, how much oxygen is available in those areas. And that's really what is helping control the phosphorus release or staying in the sediment, the retention. Great question. But usually when I talk about this work, I'm not speaking to a specific group in a location, right? Because with the pandemic, we've only been doing all this online stuff and whatnot. So I thought I would go ahead and pull out some of the Wisconsin lakes that we looked at and their bloom metrics in particular. Since we're right here on the shores of Mendota, Monona, and Wingra, right? So even though most lakes weren't getting worse, Mendota was not one of them. Its bloom magnitude has been increasing. Its bloom duration has been increasing. Severity hasn't been. Wingra, the magnitude has been increasing. And in Monona, we're not really seeing significant trends, but some evidence for increasing. But then up north, on lakes like Aliquash Lake, which is up in Vilas County, we don't see a change in magnitude, but severity and duration are decreasing. So we definitely are seeing a lot of variability lake to lake and regional differences, just thinking about northern and southern Wisconsin, right? In these bloom trends. And that's part of the puzzle that we really want to work on solving is why is Mendota getting worse and Monona doesn't seem to be changing much at all, right? Why are these two lakes that seem to be having the same things happen to it in terms of climate and the watershed not changing in the same way? Yeah.
Some lakes have a problem with acidity and some lakes don't because of the underlying rock.
Mm-hmm.
Could that be part of it?
That definitely could be. Yeah. And as we also think about recovery from acid rain over the past couple of decades, right? And how that might be changing. And that actually is sort of the line that we were on as well. So we have these 323 lakes and we were asking, what other pieces of information can we pull together to try to understand why these lakes are responding differently to seemingly the same drivers changing. So we looked at things in the way that the sediment and the water column are interacting with that phosphorus release, how those lakes are connected to each other in the landscape, how available nutrients are, and what the trends in precipitation and warming during the summer months are. And we used a statistical model to look at this. It was a mixed effects model, but in other words, we're trying to consider all these various predictors, and we're also letting them vary by region. So the same things might not be acting in the same way in the Midwest as they are, say, in Maine in the Northeast, right? So allowing it to vary by region.
Doesn't Mendota have a huge agriculture increase that Monona does not?
So Mendota certainly does have a large agricultural input from the Yahara River watershed, as well as a bit from Pheasant Branch. And there have been some changes in that land use over time. Mendota also drains into Monona, right? It's a part of that Yahara chain of lakes. So while the Monona watershed itself might be a bit smaller, it also shares the Mendota watershed in that sense because it's receiving all that water. So yeah, great question. So what did we find? Well, we found that bigger lakes are more likely to be increasing in their bloom magnitude. That's kind of interesting, right? So the bigger the lake, Mendota is certainly a little bit bigger than Monona and some of these other lakes. That might be part of it. And we found that lakes that were experiencing extreme precipitation, in other words, more intense precipitation more frequently, were likely to be decreasing overall, except for one interesting interaction. Ooh, green lakes, or lakes that are already pretty nutrient rich and experiencing algal blooms on a regular basis, if they are also experiencing a trend in extreme precipitation, so in other words, more frequent and bigger storms over the years, then they're going to be increasing in their bloom magnitude. So lakes like Mendota. But lakes where they are also green, right? So thinking about this side of chlorophyll A, these high values, lakes that are also green, but are seeing less intense precipitation over the past couple of decades are actually the ones that are getting substantially better. And so it's really this interaction between the lake's nutrient status and what precipitation it's experiencing that helps us maybe start to tease apart why we see so much variability among lakes. And just to give you a sense, because chlorophyll A values might not mean anything to you, that's a Secchi disc, which is a tool we often use in limnology, suspended 20 centimeters below the water, and you can see the different chlorophyll concentrations and kind of what that visually looks like. Alberto Secchi invented it, Secchi. All right, so thinking about what does that maybe mean for here in Wisconsin and trying to understand some of those patterns, we certainly are seeing an increase in our annual precipitation and our extreme precipitation during the summer. And that might help start to explain some of these differences that we're seeing, right? And that why lakes like Mendota, which are already very green, are getting worse. But as I said, there's still a lot of work to be done here to tease apart with these hundreds of lakes. And we need more scientists on board to help us out with that. So how can we use this information to understand how we might better manage or be stewards of our freshwater ecosystems in the face of this global change that we're seeing? Obviously, there's these complex interactions of nutrient loading and the increased precipitation, which is a part of climate change, across scales that's making uniform management. Like, we can't just say, oh, you just do this one thing and all lakes will be better, right? A uniform management solution. But in those lakes that are seeing extreme precipitation increasing, that's probably modifying how the amount of nutrients coming in and when they're coming in. And so part of that might be, for example, in the form of erosion. So erosion management might be a way to address that. Similarly, when we're having storms come in, with those storms also comes a lot of wind, and that changes the stratification or the layers in the lake in terms of their temperature and can cause mixing. And that mixing can allow the phosphorus that's down in the sediment to become available now all of a sudden to the algae that are hanging out up at the surface in the light. And so we might, it could also be that these increased precipitation and big storm events are also changing stratification and how much those sediments are being kicked up and releasing that phosphorus. And we certainly have evidence of that from some work that we've done when the derecho passed back last year. You might remember in August of 2020, the big derecho, we were measuring phosphorus release in the bottom waters of a reservoir and found that it increased substantially following that derecho almost immediately. So these storms can have a really big effect on nutrient availability. And so one thing though to take away from this is that when we're managing these really nutrient-rich lakes like Mendota that are experiencing increased precipitation, we may just be managing to run to stay in place. In other words, the extreme precipitation is working against whatever management we're trying to do to decrease blooms, and additional effort is going to be needed to overcome that precipitation. So even though we found over 300 lakes, are not, it's not widespread. We're not seeing this large increase in harmful algal blooms in hundreds of lakes like we had predicted because things are getting warmer. We do still get algal blooms in some lakes, right? They're maybe just not getting worse. And so another part of my group's research is trying to understand, well, can we predict a bloom before it happens in a given lake so that we can perhaps mitigate the consequences of that bloom? And so that's some whole lake experimental work that we've been doing up in northern Wisconsin that I want to share with you now.
I have a question about phosphorus control.
Of course.
I certainly heard a lot about it recently. Are we actually improving our situation with controlling phosphorus getting into the lakes?
That's a great question. And I think in individual areas and on individual plots of land, most likely there are areas where people have put in restoration or best management practices to control that phosphorus. But overall, we're not seeing that happen on a widespread scale. A lot of that is because nutrient management is still voluntary under the Clean Water Act, and it's not mandated. So steps in the right direction, but not big strides.
The landscape people, are they with you or against you at this time?
What do you mean by landscape people? Before I decide who's my
People
enemy.
people who come around and keep our lawns green and fertilize them.
Oh, sure. Yeah, so there are certainly things that we can do individually as landowners, right? And thinking about when is best to apply fertilizers, if fertilizers are even needed. So many of these companies can do that in a green way where they'll actually test the phosphorus in your lawn, right? If it's actually needed to be added. But if they're just trying to sell you a product, sort of that's like lawn snake oil. So that certainly doesn't help, but that's just one piece of the puzzle, right? If we think about lawns, there's all different types of land use and land ownership and management. And it can't just be small little pockets in the watershed. It has to be everybody working towards that goal. Yeah. So when I say, what do I mean if we could predict? So if we think about, this is chlorophyll concentrations in the lake over time. What if at that point, we're not predicting, the lake's already blooming, it's green, it's scummy, no one wants to go spend time around that lake. What if we knew something based upon the dynamics or how chlorophyll is changing before the lake is blooming to help us predict that a bloom is about to come? And that's really the crux of this research. And many folks have spent time trying to figure out how to predict algal blooms. And the approach that our group and collaborators are taking is thinking about them as regime shifts.
There's what?
Regime shifts. So what is a regime shift? It's a sudden transition from one stable state in the system to another. And that transition occurs because of a loss of resilience. So what do I mean by stable state? Some examples are in a grassland that transitions to a shrubland. Those are two states that ecosystem can be in, and they go through a rapid transition between the two.
How are you defining rapid or sudden transition?
Yeah, so it's not a linear increase over time, right? We essentially have, oh, I wish I'd put that slide in, where this is a whiteboard, right? Is there a marker? No, okay. So where even, let's say the driver is nutrients. You have increasing nutrient concentrations in the water and the system is staying in this clear water state. And then all of a sudden it flips up into this algal dominated state. And a bloom is a good example of that, where things stay pretty low and then all of a sudden a bloom comes on and you have that rapid growth instead of just sort of a gradual increase over time.
But like when you're talking about from grasslands to shrubs, how long a period of time are you talking about?
Yeah, so in that system, so blooms we're talking about within a summer, right?
Okay.
In that system, we're talking about over a couple of years.
Okay.
Yeah, great question. So the transition is in reference to how fast the ecosystem moves, essentially.
That's more than a couple of years.
But anyway,
Knowing something about the evolution of the ecosystem, what if you would like Mendota to be a nice meadow? Then wouldn't you be encouraging algal blooms?
I'm not sure I quite understand your question.
I'm being scientifically facetious.
Okay. I kind of thought so, but it's bad for the speaker to presume. So
Because there's a natural evolution of things. And in order for us to keep our lakes, we have to fight these forces.
Sure. So thinking about something that we often refer to as lake succession, right? So we have this glacial process that created the, Yahara Glacial Lake, the lake drained, we have these basins, Mendota, Monona and things. And yes, over geological time, they will be filling in, right? And eventually become shallower and shallower, and then could become a terrestrial ecosystem. That's on the time scale of more like tens of thousands of years though. Yeah, absolutely. And you make a good point. Time and time scale are important, right? Absolutely. But the thing about these regime shifts is they're difficult to predict, but the other state, so for example, algae coating a coral reef, it's usually not very desirable, right? And so being able to predict or prevent them is pretty important for ecosystem, and in some cases at times human health as well. And so An example of one of those rapid transition is from the clear water state in a lake to an algal dominated state, where we have lower nutrient loading in the clear water state and higher nutrient loading in the algal dominated state. Sometimes we refer to that water as turbid or not very clear. And the way we can think about how this regime shift works and how the rapid transition occurs is sort of by this diagram here. We refer to this as the cup and ball diagram. Let me explain. So the two cups here present the clear water state and the algal dominated or turbid state, as you can see there. And there's this saddle between the two that is the resilience in the system. And the steeper the walls of those basins, the more resilience there is in the system. You can think about the lake as being the blue ball that you see in the cups. So right now the lake is sitting in the clear water state. The basin walls are pretty high. And what is directly influencing the resilience is the nutrient loading, the nutrient concentrations in that system. So in this case, nutrient loading is low. But obviously these things aren't static. They don't stay the same way all the time. Maybe something like a big storm comes along and moves more nutrients from the land into the lake. And when that happens, you can think about that storm as a disturbance or a perturbation that's pushing the ball around in that basin. And so the basin walls are high though, the ball's not moving very far. And if we were to follow the ball over time, let's say measure chlorophyll in the water, these rain events and things, they would cause some variability, but it wouldn't be a lot because the basin walls are so steep, the ball doesn't move very far. And another variable we can look at, autocorrelation. In other words, how much does chlorophyll from yesterday look like chlorophyll today, right? Would also be pretty low. But let's say over time, land use changes, there's more nutrients coming into the system on average, it's lowering the resilience. And when that same disturbance, that same storm comes along, it's pushing the ball around in the basin. But now the walls aren't as steep, they're much shallower, and so the ball's moving even farther. It's taking it a lot longer to come back down to the middle there, right? And so again, if we're measuring that over time, we'd see that there's still that variability because of the storm event, but the variability is getting much higher, right? Because the ball is moving farther and farther away from the mean or the low point in that basin. Similarly, autocorrelation is also increasing. Eventually, nutrient loading is so high in the system that that same small storm event can come along and there's practically no resilience left. And there's that quick transition or flip, the regime shift into that algal dominated state. And so while we use this as sort of a conceptual model, this model is actually based on mathematical theory, that this is how regime shifts work, or what's referred to in this case as a catastrophic bifurcation. So we know mathematically and theoretically this should work. And it's even been shown in simulations and in some controlled laboratory studies. But what we wanted to know was, okay, can we actually apply this theory that there are these statistical early warnings, which are an increase in the variance and an increase in the autocorrelation before that transition occurs? Can that actually serve as an early warning of a harmful algal bloom? Well before the bloom actually happens, the transition occurs, that regime shift. And we wanted to know if we could do that by taking daily measurements of chlorophyll in a lake. So again, we wanted to know, are there statistical early warnings of a harmful algal bloom, a rise in variance and a rise in autocorrelation? And we tested this by using some lakes up in northern Wisconsin and doing whole lake experiments. And this was not to manipulate an entire lake and measure things about it. That's not something you can do alone. So this was a large collaborative effort between the University of Wisconsin, the Carey Institute, and the University of Virginia. And these are a number of the folks that have been involved and led this project. So these lakes located up in northern Wisconsin, they're actually a part of the University of Notre Dame's Environmental Research Center. This is one of two places in the world where you can do whole lake experiments unfettered. And so this has been an area where at the University of Wisconsin, we've actually been doing whole lake experiments on these ecosystems, in particular Peter and Paul Lake, since the 1950s. And that's one of the things that's made us a leader in limnological research. So what did we do to Peter and Paul Lake from 2013 to 2015? Well, in Peter Lake and in Tuesday, which is another nearby lake just up over the hill, we did daily nutrient additions. In other words, we wanted to start lowering the resilience of that system slowly over time. And as we did that, we had these sensors in the water, like you see in the middle of the screen here. And these sensors take measurements every 5 minutes of chlorophyll concentrations. So much better than doing it yourself every 5 minutes, right? And they're helping us look for those statistical early warning indicators that chlorophyll, the variance in chlorophyll concentrations are rising and the autocorrelation. And even if we saw those statistical early warning indicators, we continued to add nutrients because we wanted to know, was it actually warning us of a bloom occurring, right? Not just some other variability we happen to pick up in the system. We did the same thing in Paul Lake, except for we didn't add nutrients. We just monitored chlorophyll. It was like a good control. Very And so I think I'll leave it there, and I certainly look forward to your questions. But before I do, I just wanted to take a moment to also mention that we have a new project that's been funded by the National Science Foundation to be doing more work here in Dane County on these very nutrient-rich, scummy algal bloom waters. And we'll be looking for volunteers starting in summer of 2020 to potentially be helping us with some of that work. And so if you think it's something you may be interested in and you just want to be informed, I'd encourage you to send me an email and we'll have more information as the summer starts coming up about what those opportunities are. All right. So thank you so much for your time and I'll look forward to your questions. Yes.
I did some work with the Upper Mississippi River Commission using students from Iowa University. So would I be a good volunteer for this?
Absolutely.
Because we did water sampling, water quality sampling.
Absolutely. All we ask for is excitement and passion. And if you come with some skills built in, that's good too, but we'll teach you what you need to know. Yeah, great question. Yeah.
When you were looking at, I'm sorry, the early indicators?
Yeah.
Do you know why some of them were only 6 days and some were 30 or 40 or 50 days?
Yeah. So we were looking at a few different variables. I mentioned the chlorophyll and the phycocyanin. In addition to that, in those years where we didn't get a very big bloom, it was just pretty small, the magnitude was small, the warnings came much closer to the bloom. On years where we made it pea soup green, that's where we were seeing the bigger or the longer lag time, right? So we were getting warnings 30, 40 days beforehand. And that's actually something that we're working on investigating now, is how sort of the magnitude of that shift in the bloom is related to the resilience and the warning that we get.
And can you tell, when you look at the warnings, whether it's 6 days or 50 days until the major bloom?
Not in the way that we currently have essentially our algorithm set up, but that would definitely be something to investigate. It's a good PhD plan. Come do a PhD. It's a great question. Yeah.
Lakes can vary quite a bit. For example, Pelican Lake is a relatively big lake, but it's not a deep lake. And there's very little evidence of thermocline, and it experiences algal blooms. Where Pence Lake is a smaller lake, but it's a deep lake and has a strong evidence of thermocline, and it's spring fed, and it doesn't experience very many algal blooms. Where you think a small lake would, but it doesn't.
Yeah.
Because I think it has more resilience, right?
It's most certainly could. I would also be interested in knowing what the watershed, so sort of what's happening in terms of nutrient
The
inputs.
watershed for Pence Lake is mostly forest.
The
The agriculture is pretty far away. Where Pelican Lake, there's farms nearby.
Yeah, so it's potentially getting more of that input from the watershed, and then in that shallow Falcon Lake, correct? That's where you can have a lot more, not necessarily more phosphorus releasing from the sediments, but the phosphorus that is released is more likely to encounter algae that are up in the sunlight in the surface waters that can use it to grow.
And the storms,
And in a deep lake, you just don't get that, especially
and the
beach.
storms that go across this large lake, you get good wave action.
Yeah,
Whereas Pence Lake is kind of guarded
yeah,
by tall timber.
absolutely. And that plays a role too. So how sheltered from the wind also influences that stratification or that thermocline. And it's also influencing how much of the sediment in the shallows is getting kicked back up or resuspended. I have a student who's been working on this and trying to understand how that plays out in terms of phosphorus availability. And it is messy, but in general, in those shallow systems, higher. Yeah, so that could definitely be what's feeding the blooms in that particular lake. Also interacting with those storms, but in a different way that's not external nutrient loading. Yeah.
I'm just curious with Peter and Paul, I assume they don't have springs and they're not connected with water-wise.
Yeah, so great question. So Peter and Paul have a very small watershed. In fact, like this is the top of the hill for Peter. Peter and Paul actually used to be one lake with these two basins. And then Art Hassler in the 1950s built this berm in the middle. He called it Mary. And so Paul actually does drain into Peter, doesn't allow any fish or anything to pass through, but that's also out of design as Paul is slightly higher in the watershed, so that we're never getting, the nutrients wrapping to Peter sloshing back over into Paul.
And you know, it's like, this is northern Wisconsin, was this like, are these kettles or these glacial features?
Yeah, great question. So they are kettle lakes. Peter is about 2.5 hectares and is 18 meters deep. So essentially it's a very deep, small hole in the forest. Paul Lake similarly goes to 16 meters. So these two very deep glacial kettle lakes.
What kind of fish are in
Yeah, so that's actually one thing that's been manipulated over the years, but it's largely largemouth bass and then pumpkin seeds and some shiners that we've added over the years. So some of the original experiments looking at whether or not the food web can be manipulated to control algae happened in Peter and Paul Lake back in the 80s. So back to your original question.
They're free of zebra mussels?
Yes. Yeah, these are a very protected research station. And so there's a lot of rules about what we can bring in terms of equipment and things, because we're attempting to prevent invasive species in these waters. Yeah.
I'd be surprised you don't have zebra mussels everywhere, but I actually live on Green Bank,
Mm-hmm.
and I've observed my backyard for almost 30 years. When I first moved to Green, I was Egg Harbor, okay. First moved there, the water was awful.
Yeah.
It was gooey, and it now has near tropical clarity. I also remember huge piles of zebra mussel shells on virtually every shore. And of course, if you dive in these lakes,
Mm-hmm.
they sink to the bottom too.
Yeah.
I don't know what question I'm really asking, but how come?
Yeah. So certainly things like zebra mussels getting into lake can really shift and what's happening with the algae and the algal blooms. And we've even seen that on Mendota here, right? Since the zebra mussel invasion, we've seen changing when those blooms occur, how big they are. We have something called the spring clearwater phase in our lakes here. I don't know if you all have experienced that, but it's about in early June. You could go out to Mendota or Monona and you feel like you're in these tropical crystal clear waters. You know it's coming, but right? And that's because the Daphnia, those cows of the lake, their populations have boomed at that point, and so they're grazing everything down. But the zebra mussels have really changed the dynamics. I was just looking at this the other day because I was interested for Mendota, because we have these beautiful, just decades of data on it. The peak of the algal bloom in Mendota used to occur much later, and now we're seeing the higher chlorophyll concentrations way earlier in the season. And it has been pretty much since the zebra mussels have invaded. At the same time, though, we've also had spiny water flea or Bithotraphes invade on Lake Mendota. They are eating a lot of those zooplankton that are good at the grazing, right? And at the same time, they can't be eaten by a lot of the fish because of their long spine. And so that has also messed up sort of that grazing dynamic, and the zebra mussels are messing it up from the bottom up, right? And so the dynamics of when blooms happen in Mendota have really been shifted by these invasive species. And so my dream for the next few years, if we can get the funding, is to really put together piece by piece for these hundreds of lakes, what those invasion dynamics are, the restoration, all sorts of things to help tease apart this puzzle a little bit more. We have the trends, we know what they are, and I really want to know more about the why. So.
If you're having earlier bloom, does that mean there's more blooms?
Yeah, that's a good question. So I'm not sure necessarily we're seeing more blooms, although certainly we're having a nice warm fall, and like when Monona was pretty pea soup last week. I don't know if y'all had seen it, right? And so I think we're definitely shifting when blooms happen, and potentially, I don't have the data to back this up or whatnot, but I could see where moving blooms earlier in the season would allow for more late fall blooms to occur, just because the nutrients used up by those algae, they die off, that decomposes and releases the nutrients again. So certainly potential, but that is a hypothesis. Yeah.
So you talked about small lakes. Lake Erie had a really bad bloom, was it 2 summers ago? And it screwed
Yeah.
up the water for Toledo and such. How does this work there on a big lake like Lake Erie?
Yeah, so we specifically did not include the Great Lakes in our analysis. And part of that was because they are one big ecosystem, but need to be measured in many, many places. There's a lot of monitoring stations around them. And yes, Erie does experience these big algal blooms. It has a ton of phosphorus sitting in its sediments and it's a shallow lake, right? And so that's definitely part of what is helping support those blooms. There's also the runoff or the external loading from the Maumee watershed in Ohio, right, which is very nutrient rich. And so there's been a lot of great work by scientists, Great Lake researchers showing that it's those early spring loadings from the watershed that help seed the blooms. And then there's some evidence that it's the later season, that internal phosphorus loading that might be helping sustain them as well. So you certainly, but Erie in itself, it is an inland ocean, right? These Great Lakes are inland oceans. So while a lot of the same things apply, the sort of spatial heterogeneity is a bit more than what we were willing to handle for this analysis.
And the Maumee, you drive over on the way to the east coast, it's pretty big. What's unusual about the Maumee compared to other rivers flowing?
Oh yeah, there's just that is a particularly large hydrologic input, is my understanding.
Okay.
So
Is
not
there
being an
any-
expert on that system, but
Is there any research being done on tying up the phosphorus so that it can't help the algae?
yeah. So there are a few strategies that folks have been working on. One of them, of course, is dredging. So just trying to remove a lot of the phosphorus from the sediment and making the water deep enough that you're going to encourage stratification. So not as much is going to be available. And like I said, very mixed reviews of whether or not that's actually useful. Also extremely expensive, millions and millions of dollars, and no guarantee that it's going to work. So in my opinion, whenever a lake association asks or not, should we dredge? My answer is like a no, and cautiously maybe, probably not. There's also things like chemicals, alum, phospholock, which would bind the phosphorus.
That's what I
And there's some evidence that that works, sort of in whether it's the right ecosystem. You need it in a place where it's deep enough so it's going to create sort of a seal on the sediments, as opposed to constantly be getting kicked up and resuspended and mixing. It has less efficacy when that happens, but perhaps still some efficacy. So I think when applied in the right system, there's some evidence that that might work, but it's not a long-term solution, because more sediments and more phosphorus are still going to be coming in from the watershed, right? So it's a Band-Aid. But it could be a good Band-Aid while you're working on the things that are going to take longer in the watershed too. So those are just some of the strategies. But ultimately, we got to take care of our land in the watershed. That's the ultimate answer. Everything else is just a tool to get us there.
And is that going to take government intervention? I mean, people don't do it on their own,
Yeah.
except in little small pockets.
Yeah, but I think also it's important to recognize the competing interests there, right? I don't think anybody's voluntarily trying to make their lakes pea soup green, right? But there's not, when we think about the economic and incentive structure, particularly around things like agriculture and those margins, that's probably a large impetus there. So if we can, there's some schools of thought out there that if we can change the way that those economic incentives are structured, that might help us make steps forward in controlling nutrient inputs in the watershed. So certainly, and one of the big controls on the economic incentive structure is the government currently for agriculture.
Yeah, because there are so many streams that feed Lake Mendota that come from agricultural land. In fact, there's one stream that's supposed to meander through a marsh area where it might be clean, but it got changed, so there's a channel going, man-made channel going straight through it.
Yeah,
So whenever we get these fantastic rains, the phosphorus sediments in the stream bed get stirred up and it comes down
yeah,
and feeds Lake Mendota.
absolutely. Yeah, and that shallow area up in the north of Lake Mendota by Governor Nelson State Park, it's like my new favorite place to try to understand the bloom dynamics up there, because it's like a shallow estuary that you would see on the coast of the ocean, not so much like what I would expect as a stream input to a lake. So I think it's a really interesting place, and I hope to learn more about what's going on up there. And also, I think important to note that we talk a lot about agriculture, but human changing phosphorus inputs is really important as well. And so not just thinking about row crop agriculture, livestock, massive amount of phosphorus inputs, and in urban areas, right? And thinking about how we deal with urban waste. So there's lots of different ways we can approach the problem, and it's different for every watershed.
You mentioned zebra mussels, but quagga came after and are more dominant. Have they changed anything in the equation with the algae?
Yeah, that's a really good question, and not one that I'm as well versed on. So certainly thinking about the interaction of those invasive species themselves.
Yeah, I mean,
Yeah.
I'm still in the mindset that whenever I see a mussel, I think of a zebra mussel, but then people remind me actually the quaggas have supplanted in many cases.
Yeah. And they're also filtering and such. And so I'm not as versed in their biology. But yeah, definitely those invasion dynamics. And one of my colleagues at the center, Jake van der Zanden, has spent a lot of time thinking about these aquatic invasive species and their dynamics and how they're changing lakes like Mendota and other lakes in Wisconsin. So I can ask him about it. I think he'd love that.
Anyone else? Next week, we have Great Lakes Bioenergy Report on Rebecca Smith.
Thank you very much.
Thank you.